Thank you very much, and welcome to my presentation about Utanos. So, a quick overview of what I'm going to talk about today, an overview of the package itself, and then I'll jump right into some example use cases. One simple use case for cell line monitoring, and another for endometrial carcinoma analysis. And then I'll have a brief digression on comparing copy number signatures. So, Utanos was built with shallow genomics in mind. WGS is 30x to 50x coverage of the genome, a lot of reads. And SWGS is significantly less, 0.1 to 1x coverage. And what this really means is analysis types. That's the difference. With WGS, you can get SNVs, indels, structural variants. With SWGS, you're pretty much restricted to copy number variations. The samples that you use for these types of sequencing are fresh frozen, cryopreserved, maybe proprietary solutions, RNA later or PAXgene. These matter in context of SWGS because the advantage of doing shallow is cost. If it's a lot less cost, that expands the number of samples that you can sequence and the number of applications that you can use it for. If you have also cheap samples, that doubles in being of advantage. If you're using cryopreserved only samples, then, well, your samples are expensive. Fortunately, SWGS works for FFPE as well, and FFPE is highly pervasive in the medical industry specifically, so it's the de facto sample type used in hospitals. So this makes samples readily available, and it opens up the possibility for large cohort studies for a lot cheaper. And it also makes more samples for the average study more feasible. A brief note about copy number variations, and specifically in cancer genomics. So shallow whole genome is used currently for prenatal diagnostics, but really it comes into its own for cancer genomics. Our normal cells are mostly diploid, but tumors, you see a lot more aneuploidy. That can be at the chromosome level, or it can be at individual loci across the genome, where you have copy number aberrations. On the left, at the bottom there, you see a relative copy number plot, and that's demonstrating the relative copy number differences at specific loci. It's just a ratio median difference here in log2 transformed. Moving over to the right, you see it on an absolute scale, and so then we see ploidy of 2, or maybe ploidy of 4, or ploidy of 3, and you can see that there's a lot of variability in individual bins. So what by bins I mean is individual gray points that you see on these plots. At the shallow genome level, we don't have the benefit of WGS where you can get base pair level resolution. Here you have bin level resolution. So this might be 10 kb all the way up to 100 kb, depending on how shallowly you sequenced. So relative and absolute. Another thing to take into account when doing shallow is this issue of purity and clonality in cancer. So we just mentioned ploidy. How are we going to get ploidy? We're expecting at the single cell level integer copy or integer ploidies. Ploidy of 2 makes sense, a ploidy of 4 makes sense, but we get a lot of bins between those two. We get a lot of variability, and that's a lot because of purity and clonality. Cancers are a mixture of diploid cells and cancer cells, and also a mixture of clones. So for some simple segment plots, you're expecting a mixture. You're sampling from distributions of cells, and when you're sampling shallowly, then that kind of doubles the difficulty. So it's an interesting statistical question, an interesting algorithmic question, but it does make for variable copy number profiles. So on to the goal of this package. This is to be a common utility. This is to be composed of common utility functions that are highly accessible and easy to use, easy to modify, easy to string together, and and they sit as like the putty between the bricks of many other packages that exist in the ecosystem for making data interoperable between steps. So the scope of Utanos really begins with relative copy number calling. It takes that as input, and you can do quality evaluation and filtering, absolute copy number calling, homologous recombination deficiency identification, evaluating CN diversity. It's got many plotting options. Copy number feature modeling, which then plays into copy number signature creation and identification, and then comparing between copy number signatures. And right now, our reference genomes available are HG19, HG38, and MM10. An overview of an example workflow using Utanos. You proceed from the top left there. For segment count and median segment standard deviation, you're taking the lower end of both of those as the higher quality profiles. And you move on to segment plots. Segment plots can be interpreted at both the cohort or the individual level, where you have a probability of loss or probability of gain at the cohort level. And for individuals, you see individual copy numbers going up and down. Then at the absolute level, the same. You have individuals going up and down, or you can see it in diversity plots, where you have many, many samples stacked up against one another, but visualized as a heat map, so it makes for easy vertical comparison. Blue is losses, reds are more gains, gray is diploid. Proceeding past that, you get to copy number signatures, and then you can do feature modeling and signature creation. So now I'll show some actual examples. A simple example use case, this is for someone who was in our lab who wanted to use shallow whole genome to monitor mirroring cell lines over passages. So they were using a cell line that was a good proxy for high-grade serous ovarian carcinoma, that's HG-SOC, and they wanted to compare untransduced and transduced cell lines. So this was a background of P53 knockout, so that's just a gene knockout, and then they were adding another knockout, so BRCA wild type gene versus the knockout of that gene. And they wanted to see across passages, so that's as they're letting the cells grow out and making passages of those cells, whether or not they're increasing the number of aberrations that are present or not. And that is known to be a problem in that field. If you're increasing the number of aberrations for one cell line versus another, it makes for difficult comparison down the line. You don't have equal comparisons. So the first comparison is simple BRCA wild type, untransduced versus transduced, and it looks pretty similar. So good news for that researcher, she was happy. And compare that to the knockout, and it again looks pretty similar. And by eye, it does look quite similar. At the relative level, or using a diversity plot, it again quite similar, but it is much, much easier to compare using a diversity plot. And specifically on chromosome 12, you can see at the beginning there was a little bit of an increase for some gains there. And that was really the only thing to take note of. What she particularly found useful, though, was that the oscillating copy numbers, what we might in the business call chromothripsis in chromosome 2 there, that was specifically reserved in the same form across all of the passages. And so that was very useful for her as well. So that was a simple example. Now onto a cohort level analysis. A brief note first about P53 abnormal endometrial carcinoma. That's the main subject of this study. That's one of the 4 that was identified several years ago in a few seminal papers that split endometrial carcinoma into molecular subgroups. One was MMR, another was POLE mutated, another was P53 normal, otherwise called no specific molecular profile, so it didn't have a molecular signature, and then P53 abnormal. P53 abnormal is the most aggressive form, and it encompasses roughly half of all mortality from the disease. And so that's why they wanted to study it, and they sequenced 196 samples using shallow whole genome sequencing. On the bottom left there, we have the plot of all those samples. And the first step to creating signatures was to model some features. I'm just going to highlight segments as something that we can keep in mind as we walk through these plots. So the segment lengths is just in base pairs. So this might be 5 million, this might be 30 million. It's just a segment of the genome. So we're going to model that. Using a mixture of either Gaussians or Poissons, depending on the feature involved. And then we're going to take those samples and calculate a sum of posteriors vector for what the probability of each sample was belonging to each component. So each component is each individual curve. We then use non-negative matrix factorization to decompose that into a component by signature matrix and a signature by sample matrix. So for those of you familiar with the computational difficulties here, what we're really trying to figure out is the rank of the subsequent matrix. So the component by signature matrix, we've got a rank of 5, and so the optimal number of signatures here is 5. So one of the findings from this study was that they identified 5 copy number signatures that were able to that were identifying of certain things within their population and stratified the samples. If you want a little bit more detail on those findings, you can see the poster that's in the session that it was actually today. So you can examine this plot in a bit more detail. But briefly, you can co-plot signatures with clinical indicators like survival, histotype, stage, HER2 status. And very briefly, you can see that the ploidy was specifically associated, high ploidies were specifically associated with signatures 3 and 4. And that's easy. That comparison is easy to make with the top bar versus the middle blue and green section. So a related disease to P53 abnormal endometrial carcinoma is the NSMP that I mentioned. And with Utanos, just adding that NSMP group in with the P53 abnormal group was quite easy. Of note is that I was able to make all of these plots over the last slide and this slide in the course of about 45 minutes. So it really, really helps for that kind of analysis. So I wanted to add these samples in with the last group. So just stack them in at the bottom there. The NSMP now is in the black, and you can see, as expected, NSMP is a lot more copy number normal. So there's a lot more diploid there. There isn't quite the massive high copy number events, the 4s, the 5s, the 6s. And then you can show the signature exposures for that NSMP group to the signatures that were made using the P53 abnormal group to see if those samples that are somewhat diploid looking are similar for P53 and NSMP. So blowing it up a little bit, you can see that it's signature 3 and signature 4 are elevated for the large copy number gain group. And then for the more copy number normal group, it's signature 1 and signature 2. And it's the same for both NSMP and P53 abnormal. It's a relevant finding for that for those researchers. A quick digression on copy number signatures. Specifically of interest to a lot of computational folks is how similar the signatures are between the different cancer types. So there have been a few different signature sets published over the last few years. The first group was McIntyre et al. In 2018 for high-grade serous ovarian carcinoma. And then earlier this year in Jameson et al. 2024 for P53 abnormal endometrial carcinoma. So if we wanted to compare between those two signature sets, Utanos could be used to compare using Wasserstein distance, either simple component plots or alluvial plots, and how samples are segregated based on signature exposure. You can just see the samples flowing from one signature set to the other. The Wasserstein distance is just Earth mover's distance, so transforming one curve into another curve. How much weight and how much cost is there to transform one curve into the next? So that is everything for Utanos. Some acknowledgements, of course, all of my co-developers and the wet lab analysis folks that were involved in the P53 abnormal endometrial carcinoma dataset. Some user recommendations, cell lines from Dr. Ian McNeish, and advice, funding, supervision from all of those folks. So thank you very much for your attention.
Thank you very much for that. We have time for questions.
Hi, great. Thanks for talking. Say hi to David Huntsman when you get back.
Well
the behavior of different cancer types with respect to their genomic abnormalities will vary. So this will work for where copy number is involved and so forth. Are there any cancer types which would also fit for that would generate circulating DNA?
Yeah, so ctDNA and cfDNA are definitely of interest here. Those profiles fit quite nicely in with this. So there are people in labs that I'm directly associated with that are interested in using Utanos for cell-free DNA. Okay. That work is going on now.
Great, thank you.
No worries, thanks. Is that one on? Nope. I think the button's at the bottom.
Thank you. Thanks for the great talk and the great tool. So when calling copy number variants with high coverage panels or whole exome sequencing in cancer, usually it's important to take into account the purity of the sample, right? So purity 15% or 30%, it makes a big difference on variant on copy numbers. Could you please elaborate how is it with shallow sequencing? Do you take into account purity or it's
Absolutely, yeah. It's a big thing to consider. So if I move back to this plot closer to the beginning of my talk. So here you can see the scaling of relative to absolute, but we start considering purity pretty early with the then quality filtering that we do as well. So when we solve for an absolute copy number profile, we assume specific cellularities and specific ploidies, and then we calculate mean absolute deviation for probability of that ploidy, and then the lowest one is the one that we choose. That's one option though. You don't have to necessarily choose your ploidies that way. You can also integrate targeted panel sequencing, and so if you use targeted panel sequencing, then you can use the variant allele frequencies and use that as an approximate of the cellularity of that tumor. So that's another option. Or if you happen to have done FISH on some of the samples or some other modality to find ploidy, you can also use that. And Utanos actually has functionality for each of those cases where it will scale samples depending on that. So for samples that are just too low cellularity, too non-clonal, you do get very, very messy profiles. And in those cases, shallow whole genome just doesn't have enough information present in order for you to scale to the ploidy that is the true ploidy. And in those cases, it's tough to really say that that is the true ploidy, because the true ploidy is really a mix of diploid and tetraploid and quadruploid. And in that case, maybe shallow whole genome just isn't the right modality that you should be using. You really need to maybe section that tumor a little bit differently or study it at the single cell level, because you need more information to resolve it. But we found pretty good results with using just the mean absolute deviation for scaling ploidies. For relatively clean samples, I think we did a test with FISH, and I think for 17 out of 18 samples, the ploidy was scaled accurately. So it was pretty close. Yeah. No worries.